NIMOTUZUMAB is a monoclonal antibody for therapeutic use with proven clinical safety and efficacy, used as monotherapy and in combination with chemotherapy and/or radiotherapy. It obtained its first health license granted by the Center for State Control of Medicines, SACMED, in 2002 for treatment of advanced head and neck tumors. It is also currently registered for use in highly malignant nasopharyngeal, esophagus, pancreas, non-small cell lung tumors, as well as glial tumors of high degree of malignancy for pediatric and adult population, and more recently for post-COVID-19 pneumonia in severe or moderate patients. This product is manufactured at the Anticuerpo plant, one of the Molecular Immunology Center production facilities, which was built in 2009. In 2011, the company obtained the Good Manufacturing Practices certificate from SACMED for the manufacture of NIMOTUZUMAB's active pharmaceutical ingredient. The building is divided into two zones with a Class D controlled environment in the production facilities, which allows for the production of the active pharmaceutical ingredients of two monoclonal antibodies simultaneously. The plant is equipped with perfusion-stirred tank fermentation technology for superior cell culture, a 50-liter inoculum bioreactor, 500 and 2,000-liter production bioreactors, harvest filtration systems, preparative chromatographic systems for purification, refrigeration chambers, culture media preparation and filtration system for culture and solutions, and sterilization equipment. Large volumes of liquids, up to 5,000 liters, are handled using disposable sterile plastic bag systems and stainless steel storage tanks, ensuring low microbial load in line with this type of production. The manufacturing process of the active pharmaceutical ingredient begins with the unfrozen of a vial from the working cell bank. The culture is first grown in stirred flasks. When the required biomass is achieved, it is transferred to the inoculum bioreactor. The culture is further multiplied, and once the required cell mass is reached, the production scale bioreactor is inoculated. The harvest obtained from the production bioreactor goes through a purification process which consists of several steps. Samples from the production process, as well as those that guarantee the quality of auxiliary production systems, are delivered to the quality control laboratories, accompanied by the request for the tests to be performed. The quantity of samples received is sufficient to perform the whole set of tests at least 3 times, but only the sample required for one analysis is distributed to the analysts. The rest is properly stored as a control sample for use in case of complaint. If the results are not as expected, an investigation is carried out to determine whether the problem is inherent to the sample or to the undertaking of a test. The SIM quality control laboratories are responsible for evaluating the quality attributes of each molecule. Among the tests performed to the samples are identity, which is the determination of the isoelectric point, purity, strength, which is determining the concentration by spectrophotometry. The levels of contaminants associated with the process are also evaluated, such as the concentration of residual DNA, host contaminant proteins of the cell, and protein A content that may be released during the purification process. Purity is determined by HPLC molecular exclusion chromatography and electrophoresis in polyacrylamide gels on the reducing and non-reducing conditions. The formulation process aims to adjust the protein concentration through the addition of formulation solutions, The active pharmaceutical ingredient is stored in disposable, sterile, non-pyrogenic 3D flexible bags. The temperature and the time the bags contain the active pharmaceutical ingredient are critical and controlled during their storage. The pH, conductivity, endotoxin, and polysorbate 80 content of the formulation solution are the critical parameters that define the quality of the product and, therefore, of the stage, as well as the stability of the final product. On the other hand, the aseptic filling process of the formulated product starts with the sterilizing filtration by a 0.2 micrometer membrane immediately before the filling process. The filling, stoppering, and capping operations are performed under air laminar flow Class A and under a Class B environment using the vials closing filling machine with a capacity of 4,000 vials per hour for a maximum batch size of 10,000 vials. The batches already approved in the visual inspection process and released by analytical control are delivered to the automatic labeling area. The labeling machine works under the thermal printing principle with a capacity of 9,000 units per hour. Then, final packaging is performed according to destination and customer's request. The product is stored in refrigerated quarantine chambers between 2 and 8 Celsius degrees until it's released for marketing. The finished product is only distributed when the Quality Assurance Department issues a certificate of compliance with good manufacturing practices. The Molecular Immunology Center has other manufacturing sites. One in Beijing, China, a joint venture that develops and commercializes more than 100 kilograms per year for the Chinese market, and a joint venture based in Singapore for the development and commercialization of NIMOTUZUMAB in Europe and among members of the Association of Southeast Asian Nations. Currently, 2 technological transfer projects for formulation and final processing are being carried out in Turkey and Indonesia, among others. With them, technology transfer has been carried out for the manufacture of the active pharmaceutical ingredient.